I am Coral or Dar, Dolly. I go by a lot of names for my real name, but let's just give you my real name. It's Dar. Hi, I'm Dar. I am an aspiring scientist. I'm about to graduate. Fingers crossed. And I want to go into water quality databases research. So let me just spill out my brains. So I want to do water quality research on eutrophication. So that would be the overnutrition, so an abundance of nutrients like nitrogen and phosphorus in the water that would not normally be there in an oligotrophic, which would be a nutrient poor, like seawater environment. So, so I want to research eutrophication. I want to research it using computers. So I want to use databases and I want to use data and just accumulate all of it. And my goal is to be a water quality scientist, examining plastics and nutrients and their effects on ecosystems such as coral reefs, um, their associations with algal blooms, let's say, and how that associates with coral reefs and other environments. So that is my career goal. So now that we've got that out the way, I'm now going to talk to you about the random assortment of research I've done in my entire undergraduate career. So, number one, freshman year, I was super into sustainability. I still am, but I was super into it. Like I was very granola. Like I loved sustainability. Like I would yap about it all the time. I would be that one person in the friend group that's like, stop using straws and instead use bamboo and stuff like that. Like I was the one that was always reposting that and sending it. I was the person in the family that was always like, why are we buying these sponges when we can buy those sponges that are natural and they're made out of loofah, stuff like that. So freshman year me, coming into college, I'm very granola. I take a lab. It's during COVID, during the outbreak. I take a lab. And we have to take it online. So I'm like, you know what? If this lab can satisfy my prerequisites and my requirements, let's take it. So what do I see? A coral lab. Oh my god. I love coral. I thought so. So, as a kid, my dad would always have Animal Planet on. And also, sorry if you see me waving stuff around, this is a poster that I'm going to be showing you. So, when I was a kid, my dad always had Animal Planet on. And when we couldn't afford cable, which was 90% of my childhood, I would be at my aunt's house. And my dad would come to pick me up after a while. And I would just be hanging out at my aunt's house. It was not because of the cable. But my dad would take advantage of the fact that they had cable and he would put on Animal Planet. And I have a distinct memory of my dad sitting in the recliner chair, all the way reclined, relaxing because he just came off of work. And my dad's always been a construction worker. He would work 12-hour shifts or more. He was always on duty pretty much like all the time. Like anybody would call him anytime, even when he was off work. So you can imagine a construction site is a very stressful environment. There's a lot of hazards. There are people dying sometimes. That has happened. My dad has either seen it happen or heard of it happen at his site. Thankfully, not when he was in charge, because he wasn't in charge at the time. He would later on become a superintendent. But at the time, my dad was just a regular construction worker. And so he would be very stressed out, working very long shifts. So, what do you want to do when you do that? Do you want to look at violent things? Probably not. So my dad always loved nature. He's Cuban, I'm Cuban, my mom's Cuban, everybody's Cuban in my family. So nature was just outdoors. You know, kids in Cuba, they go outside, they play. Neighbors know neighbors, they're not scared. So people did go play outside. It wasn't like American kids where we were more sheltered and we are more sheltered. So my dad has always loved nature. And so when he would come after work, he would go pick me up at my aunt's house when I would be over there, and he would watch Animal Planet, sitting back on the recliner, reclined all the way, and I just have this very distinct memory of my dad reclined on the recliner, very relaxed, watching a gazelle getting eaten by a lion. So childhood me thought that was really weird. How do you not feel anything for this animal? And then I was like thinking and watching my dad's expression, and it wasn't yeah, go kill that gazelle. Or it wasn't a no, don't kill that gazelle. That's so sad. It was more of a wow, that's the circle of life. That is nature. And that in itself, although there's strife, there's toil, there's torment, that's beautiful. And me as a child observed that facial expression and observed what was on the screen, and I was like, that is beautiful. And so I would always have this appreciation for nature, which led to me being very granola, loving sustainability, and all of that. So when I saw this coral lab, I was like, I've always loved nature. As a kid, I would pretend Poseidon was pushing me around in the water and I could control the waves. So I love the water. So why not take this coral class? Because I think corals are beautiful. So I take this coral class, and when I tell you that was the climax. So when I say the climax, think back to your fifth grade English class, and they show you that little chart, and it rises up in tension, and the action punches up climax. That was a breaking point. Not a breaking point, a making point of my career. That is what propelled me forward in environmental sciences. Because at the time, I would cry myself to sleep every night in university. And I'd be like, I'm a pre med. I have to be a doctor. My family's not in the best circumstances. I have to be a doctor. I have to be a doctor. I have to be a doctor. So I took this choral class. First day there, TA was wonderful. Chillest guy you've ever met. Chillest guy you've ever met. His name was Joshua. So Josh, as we called him, was the chillest dude you have ever met. And I instantly, with my undergraduate freshman jitters, was like, relaxed. Like, this is a chill class. So I'm going to really pay attention because I like the atmosphere. I can pay attention. My anxiety's at ease. And he starts walking us through. He starts walking us through what corals are. They're cnidarian, so they're related to jellyfish. They're related to anemones. Okay, got that. I already knew that because I researched things that I like a lot. Okay. Then we get to talking about coral bleaching, and I was like, okay, I've heard of this happening, but why does it happen? So he talks to us. And we learn coral bleaching is a process that occurs when a coral is stressed. And when a coral is stressed, they expel their zooxanthellae symbionts, which are the little algae that give them color. So corals actually don't have any color. They're fully white because they have calcium carbonate skeletons inside that are white. And so they get their color from the algae that they house inside. So it's like your house being white, but there's a layer of paint on top because the people that live in there painted it. So we learned from that coral bleaching occurs when the coral is stressed because of heat or too much acidity in the water, and it goes to the guys inside the house. It's the landlord. What the hell are you guys doing? I don't know what you guys are doing here. Even though you've been helping me because you feed me, you give me money. I don't know what you guys are doing here. Yeah. And so corals can actually hunt. Well, not hunt. They filter feed. So let's say about 10% of their diet comes from filter feeding, 90% of it comes from those algal symbionts. So when the coral expels them, because it's like, what the hell are you guys doing in here? I don't remember you guys being in here. It turns white. The paint is gone, and now its food is gone. Let's say its money is gone because the landlord expelled its tenants. Okay, and I learned that I'm like, okay, fascinating. But that was cool. That was cool. Then we learned corals are mostly like a colony, a reef, is mostly one person. It's one individual with a lot of twins. It is one guy that said, you know what, I think I'm great. I'm gonna make about 20. I think that's good. I think I'm cooking here. I think I'm cooking here. And cook they do. That's how we got Great Barrier Reef. We got tons of corals, and most of them are the same guy. So let's say you got one guy in the hospital and he's sick. Not only is he sick, it's genetic. Let's just give an example. It's genetic. So the guy's like, holy, I I don't know why I have this on mind, but let's say the guy has fatal insomnia, which is a genetic condition. And the guy's like, holy, I cannot sleep. I'm just, wow, it's been 4 days since I've slept. I'm hallucinating and I'm dying. And the dude dies because there's no cure to it. Then you open the hospital doors, and then 20 people that look the exact same as him walk in, and they also can't sleep, and they're also dying. So, what I'm trying to get at here is when corals get a disease, a lot of the times, if one dude has the disease, they're all gonna get the disease because they're genetically identical. So they have the same caveats in them. They have the same weaknesses, the same strengths. They are the same person, just multiplied. So we learn about that. We learn about how when a disease hits a coral colony, it wipes out 70% of corals. That was the beginning of what hooked me in. That hooked me in. I'm like, okay, this is insane. This is like so fascinating. But then he hits with a banger. And then he goes, did you guys know that corals can actually starve? Because the little algae guys in them are like, hey, there's a lot of food outside. Let's go outside and roll over the coral because there's so much food. Just completely cover the water so it can't photosynthesize. I was like, what do you mean? How does that happen? Well, that happens. Let me talk about my career goals. That happens with eutrophication. So when there's too much nitrogen and phosphorus in the water, a lot of the times from agricultural runoff, and wastewater runoff. So when we treat our wastewater, it's clear, it's treated, but there's still nutrients in it from the waste. Let's just say nice. Let's just say the shit, the shit and piss. Yeah, the shit and piss we dump in the ocean. Yeah, it makes the algae go, holy shit, this is delicious, and they overgrow, and it makes that green goop in the water. Yeah, you heard it right. Yeah. But rest assured, most of the algae is there because of actual farm water runoff. It's not because of wastewater runoff. I did my research. I know that. Because I was writing a paper on it. So we know that. And I was like, eutrophication. That's a banger word. I'm going to put a pin in that. And years from now, I'm going to still be obsessed with it. And that's where I am today. So I am still obsessed with the concept of eutrophication. I've written, I think about 7 papers on it for 7 different classes. And I'm obsessed with it. And I want to study it for my career. So that's how I ended up in environmental science. That one coral class, I was like, being a doctor of medicine, I'm going to be a doctor of algae and water. That's what I'm going to be a doctor of. And then water break. I need to take the water break because I'm dying. I'm going to turn you guys' volume up so I can hear what you were thinking. Huh? What do you think so far? So, I'm back. Water. So, from corals, where did I go? Well, I'll tell you where I went. And I went for a while. Oh, it's raining outside. I studied economics of plastics recycling because I was still very granola for 4 years. So, 4 years. And in that time, let me tell you what I learned. So, number 1, I live in Miami. That's why I was taking Coral Lab. That's why I'm so in love with the water. That's why I love algal bloom research. I live in Miami. So, what does Miami do? Well, Miami-Dade County, like a lot of counties, it has waste. And like a lot of counties, which is to say all, like all counties, it has to deal with the waste, has to manage it. So how do we manage plastic waste that we can't just dump, right? You say to yourself, Oh, you naive little thing. So actually, we do dump it in landfills. But you know what majority of it is? It's recycled. Now that's very, very good, isn't it? Oh, what's that you say? But if all of it was recycled or majority, wouldn't you have heard about it? Oh, well, that's a good question. Actually, it's recycled using waste to energy. So, what is waste to energy? Waste to energy is when we incinerate plastic and use the small, absolutely minuscule, tiny, 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 tiny little energy that we get back from incinerating it, and we call it recycling. So let me get one of my dolls. She's gonna be the public. Um, what do you mean? We burn most of our plastic, and in municipal records say that it's recycled. Well, I'm glad you asked. Yes, we do burn most of our plastic and call it recycled. Thank you for asking. Why do we do that? I don't know. How much money are we saving from it? I don't know. I looked through the papers. It even got cited in a state commissioned University of Florida study, along with my peers. I looked into the math. I don't know. Yeah. Sophomore and junior year me did not know, and I still don't know. So, yeah, most of our plastic here gets burned. And it's called recycling so that it sets no alarm bell. Very sad. Um, you know what else we do in public? Oh, shit myself. Cut. You know what else we do public? Yeah. We used to sell our our plastic waste to other countries in landfills. So that they were drowning in garbage. And I used to sell it to them. I mean, we used to pay them to take it. Because they did not need it. Because you can't, the thing about plastic waste is the more you recycle it, the less durable it is, and you can't recycle most of it actually. So according to the county, um, yeah, you can't recycle plastic bags. You can't recycle electronics without going to proper recycling places, which most people won't. You can't recycle pressurized containers like hairspray, which most people won't recycle properly. They won't take it to the actual centers if they need to. You can't recycle batteries. I feel like most of you know that it will explode. I feel like most of people I know don't recycle batteries properly. And if you use a vintage razor like I do sometimes, you can't recycle razor blades unless you go to the proper procedure of containing them properly, sealing them, and then taking them to the county. Yourself, yeah, you can't recycle that. So all of that waste just gets put in a landfill by most people, or it gets incinerated if it's sorted properly, which a lot of the times it's not. So there you go. I'm back. And I'm using the Laguna Blue doll as my mic. Even though I don't have a mic attached to it, I'm using a desktop mic that is holding a camera, which is a webcam. So the reason this little doll is here as my mic is because I want to show you just how much I love the ocean as a kid. My favorite Monster High doll, Laguna. Who's my favorite Monster High doll currently still? Laguna. You know why? Water. She has to do with the water, and she's a little mermaid. She's a little sea creature, and I wanted to be her so bad. My second favorite, Venus McFlytrap, because she was an environmental scientist and conservation advocate. You could say I was meant for this career. Um, number three, two, three. Number three. What else did you research? How many numbers are there? I'm glad you asked. There are four numbers. Okay, so number three. Bioinformatics of coronavirus proteins and specifically SARS-CoV-2 across numerous species that focus on the NSP proteins, NSP5, NSP6, NSP11, I think, and I think also NSP14. Or was it 12? I studied a lot of non-structural COVID proteins, so bear with me, I might be wrong. So a little ASMR, removing the post-it. Oh, and my tour. Okay. Moving the post-it that has my government name on it so you don't see it. Removing the rubber band. And what am I doing? Showing you my poster of research that I conducted another event like 3 years ago. Check that my name's on it, and it is, so I have to cover that real quick. I'm 6 feet tall to get you to see me, but here, hobbling around, is my poster. So, I'm going to step out of the frame so that I don't get scoliosis. Let's start. So, I made this poster on NSV 13. I had originally checked a ton of pockets. I had also checked a ton of proteins. NSV 13 was the most viable for this internship. Before the internship, I was studying NSV 5 and other proteins. So this is more refined. This was my second go around, and I'll have you look at the introduction. So, all right. So intro, SARS-CoV-19. We already know it was terrible. We were trying to find druggable pockets, so that would be pockets within the protein structure that you can inject. And by inject, I mean bind a drug to. So that would be bind a chemical molecule, into a protein. So the goal is we want to find proteins using BLAST across several species. You can't really see it. I'm trying to show you. But the methods, we're trying to use BLAST, which is a tool that lets you see proteins across numerous species. And we were focused on the SARS-CoV-2 variants among different species. So I looked at ones that infected dolphins. I looked at ones that infected humans, of course. I looked at ones that infected rats. I looked at ones that infected a ton of stuff. So what did I find? I also found MERS and other related sequences within those numerous species. I then aligned them using a multiple sequence alignment using software like JALView, and it was aligned with the muscle algorithm. So what I did, I opened a program, loaded in all the data I had found, and I clicked the little button, and it aligned everything by the amino acid. So the amino acid are the little blocks, and the protein is when you put the little blocks, the little Legos together, and you build a little train, okay? And then from there, I said, okay, let's run it through muscle. Let's use the MAFT-Lindsay algorithm. And that's just how the professor wanted it to be done. I don't know exactly what it did. But we then built trees using IQ-Tree and Fig-Tree, which are also two softwares. And they gave us this beautiful thing. And those softwares basically showed us that widgeon, Canada goose, duck, turkey, and beluga whale, for some reason, coronaviruses were very related. Okay. MERS, Hypsigo bat, Erinaceous hedgehog, BTTP beta cove, Tylonic terrorist bat, and were the most related to each other. Then we saw SARS-CoV-2. That's our big guy that we want to look at. Bat COVID, pangolin COVID, Zarya bat COVID, and Rosettus bat COVID were really related. So let's put a pin in that. Holy, this is interesting. And then we saw human coronavirus, Batillon coronavirus, ferret coronavirus, and Rhinolophus bat coronavirus were related. And in our tree, we see the bird COVID with the beluga whale. It's split off early, so that's less related. Then we see we have a clade, so that would be these little structures, these little triangles. A clade that splits off into all of the other coronaviruses and the ferret coronavirus. So that's more of an outgroup. And then it splits off into the human coronavirus and COVID. And that's where we want to pay attention. So we were looking at that. We were like, okay, this is interesting. This is interesting. And then we want to go, okay, so those are the most related. I understand that. But I want to see where I can bind a protein to a molecular structure, like a drug, to a protein. Where can I find that inside the virus? So that's when we used something like Conserve and Primal. So we use Conserve to look at the conservation of proteins, and then we use let me see. Then we look at the conservation of proteins, and then we use statistical analyses using scripts to look at how conserved and how evolutionary, so like how evolving these proteins are. And the reason we want to look at that is because we want to look at proteins that evolve less, which means mutate less. So when you hear virus, you hear a lot of times, oh, it mutated, there's a new strain. Yes, we want something that is so integral to the virus, its structure and replication, that it cannot exist without it. So if you change one amino acid in it, it cannot exist. And if that structure cannot exist, the virus cannot exist. So that's why we want a conserved structure or protein, and we want it to be accessible to a drug. So we want it to have binding areas where a drug can come in and do its thing and bind to it. So in that, I found Pocket 13, 17, 22, and 21. And out of all of those, Pocket 13 and Pocket 17 were the best pockets. So they had the highest conservation, the lowest evolution, and their drugability rates, so the ability for something to bind to them, were above 90. Pockets 21 and 22 were of interest in that they were very accessible with the highest drugability, but they were changing a lot. So that's what I learned. I learned how to use Primal to visualize a protein structure. I used Conserve to look at the conservation of that said protein. I used IQ-Tree, Primal, Spider, Fig Tree, JALView, Muscle, Math-Lindsay, Blast. I used all of it, and I used Python scripts that were pre-written to come to this conclusion. So that was research number 3. So what the hell is number 4? Currently I'm doing number 4, and I'm mapping Florida coastlines with JS. And that's in progress, so I can't really share much about that. But that was my research journey. If you watched till the end, I love you. Thank you so much. Please give me a scholarship. I am in dire need because I don't have a way to fund my master's that I want to do in water quality and eutrophication. So I would really appreciate if you sent this to any scholarship office you saw, if you considered me for a scholarship, or if you're just a friend or a viewer of my channel, if you hit the like, hit the subscribe, and hit the bell, and I'll see you next time.